The visionary behind Gleevec, Dr. Brian Druker. Dr. Brian Druker, born in 1955 in St. Paul, Minnesota, is a physician-scientist whose relentless passion for finding better treatments for cancer led to one of the most significant breakthroughs in oncology. As the director of the Knight Cancer Institute at Oregon Health and Science University, OHSU, Dr. Druker revolutionized cancer treatment by developing Gleevec. Dr. Druker's journey began with a strong interest in science, which was evident from a young age. Despite initial setbacks, including rejections from several prestigious universities, he pursued his education at the University of California, San Diego, where he majored in chemistry and later attended medical school. Inspired by the progress in treating childhood leukemia, Dr. Druker focused on understanding the molecular mechanisms of cancer, particularly chronic myelogenous leukemia, CML. Can genetic alterations cause cancer? In the 1950s, scientists had a limited understanding of how genes influence cancer. It was a time when most investigators did not think that tumors were caused by genetic mutations, wrote Peter Nowell, MD, then a researcher at the University of Pennsylvania. Despite this paradigm, some scientists wondered, can genetic alterations cause cancer? However, they lacked consistent evidence to support this hypothesis. During that time, Dr. Nowell spent his days peering into the microscope, studying cancer cells from people with CML. Back then, patients diagnosed with CML were basically handed a death sentence. The available treatments were harsh, invasive, and not very effective. Despite the popular belief that cancer was not caused by genetic changes, Dr. Nowell was interested in the chromosomes, structures that carry genetic information, of leukemia cells. He searched through the University of Pennsylvania for someone who shared this interest, but came up empty-handed. Luckily, someone introduced him to David Hungerford, Ph.D., then a graduate student at the Fox Chase Cancer Center in Philadelphia. Dr. Hungerford was studying chromosomes for his graduate thesis, so the two decided to collaborate. Discovery of the Philadelphia chromosome in leukemia. In 1960, doctors Nowell and Hungerford examined cancer cells from two patients with CML and noticed something peculiar. One of the 46 chromosomes was abnormally short. Though they had no clues as to how the short chromosome was created, or what its function was, the genetic alteration piqued their interest. Curious to know if the small chromosome was a common feature in CML, they eagerly examined leukemia cells from more people with CML. Sure enough, they found 7 more patients who had the same abnormal chromosome. The tiny chromosome was later named the Philadelphia chromosome, after the city where doctors Nowell and Hungerford discovered it. Further research showed that 95% of patients with CML have the Philadelphia chromosome. It wasn't until the 1970s, when techniques for studying DNA improved, that researchers realized the Philadelphia chromosome forms when 2 chromosomes break and the pieces swap places. A few years later, Nora Heisterkamp, MD, then a researcher at NCI, and her colleagues discovered that when the Philadelphia chromosome forms, 2 genes that are normally separated get fused together. The hybrid, called a fusion gene, is known as BCR-ABL. Owen Witt, MD, and his colleagues at the University of California, Los Angeles, later proved that when BCR-ABL forms in blood cells, it causes CML. The birth of a breakthrough. Dr. Druker zeroed in on the BCR-ABL protein produced by the Philadelphia chromosome. In collaboration with Novartis, Dr. Druker and his team developed imatinib, later known as Gleevec. This drug was designed to specifically target and inhibit the BCR-ABL protein. The initial clinical trials began in 1998, and the results were nothing short of miraculous. Patients who had exhausted all other treatment options saw their cancer disappear, and their blood counts returned to normal. Real-life impact. Katie's story. Katie Knudsen was just 6 years old when she was diagnosed with CML. Her parents were devastated, as the prognosis was bleak. However, Gleevec had just been approved by the FDA, and Katie began treatment in July 2001. By February 2002, there was no detectable leukemia in her body. Katie went on to lead a normal childhood, excelling in academics and dance, and later graduating with honors from nursing school. Her story is a testament to the life-changing potential of Gleevec. The pioneer, Judy Oram. Judy Oram, diagnosed with CML in 1995, had run out of treatment options by 1998. She joined the Phase I clinical trials for Gleevec and became the 9th patient to take the drug. Judy has been on Gleevec longer than any other patient, and thanks to this miracle drug, she was able to become a grandmother and enjoy many more years with her family. Transforming cancer therapy. Gleevec's success in treating CML was just the beginning. The drug was also approved for the treatment of gastrointestinal stromal tumors, GISTs, acute lymphoblastic leukemia, ALL, and other cancers driven by similar genetic mutations. Its targeted approach minimized damage to normal cells, reducing side effects compared to traditional chemotherapy. A legacy of hope. Gleevec's impact on cancer therapy cannot be overstated. It transformed CML from a fatal disease into a manageable condition, significantly improving survival rates and quality of life for patients. The success of Gleevec paved the way for the development of other targeted therapies, revolutionizing the field of oncology and inspiring hope for a future where cancer can be effectively treated with precision medicine.